protein folding is the process wherein a nascent, again, a nascent polypeptide is being folded or being improved para maging native conformation siya. In other means, pag sinabi nating nascent to native, we are making a non-functional polypeptide into a functional one para magkaroon na siya ng biological role inside our body. And please note that in protein folding process, this is a highly regulated, again, this is a highly regulated process, And in addition, this is a non-random one, okay, non-random process. So highly regulated and also non-random. So bakit natin sinabi na highly regulated and also non-random? Please note that protein folding is essential para magkaroon tayo ng normal functioning inside our body, because otherwise, kapag nagkaroon ng alteration sa folding, that could result to diseases or protein diseases, which will be tackled later. In protein folding, the process of maintaining or the continuous maintenance of protein synthesis and also folding is called proteostasis. So under proteostasis, there is coordination of different processes, and these processes could be categorized into three. Again, these processes could be categorized into three. So number one, can you follow, by the way? Okay, so thank you. So for the first process, that involves synthesis of nascent polypeptide, And in other words, pag sinabi nating synthesis of nascent polypeptide, protein synthesis. And the part of the cell that produces proteins are the ribosomes, as we remember. Okay, are the ribosomes. That's right. And then after that, after synthesis, of course, folding has to occur. Kaya papasok dito ang protein folding process to make the polypeptide produced by ribosomes functional, para maging kapaki-pakinabang ang ginagawa ng ribosomes. Now, to be able to regulate well folding process in proteins, we have the specialized proteins that assist in protein folding, and these are called the chaperones. Kaya meron tayong segment ng discussion later na pag-uusapan natin ang chaperones. And then for the last, this involves degradation. Okay, degradation. How important is degradation? Degradation is important in case there will be misfolded proteins. Remember, class, kapag meron tayong misfolded proteins, this could be a threat. MF means misfolded. Okay? Kapag kung sakali na may nabuo tayong misfolded proteins, this could be a threat para magkaroon ng pathological condition ang isang tao. Or in other words, magkakasakit siya. So to prevent that, these misfolded proteins have to be sequestered or degraded. Kaya part siya ng proteostasis. Okay? So altogether, these are the different processes involved in proteostasis. So ito rin yun. Yung naka-second bullet, ito yung summary niya. Naintindihan yung 3 steps, everybody? Okay, that's right. So with this figure, this figure shows the summary of proteostasis, wherein we discuss that how many processes do we have? Ilang processes meron tayo? 1, 2, or 3? 3, very good. So first, we have synthesis of nascent polypeptide brought about by ribosomes. And the template na pagkokopyahan nun is the primary transcript or the mRNA. Okay, galing siya, of course, sa mRNA. And then, as we said, folding occurs. Okay, folding occurs so that nascent polypeptide would be converted into native proteins. Now, during the process, of course, there could be problems na pwede ma-encounter. Kaya to the rescue ang mga chaperones. Kaya every step of the way, makikita nyo na may mga chaperones na nag-act. result. And then finally, kung sakali na hindi naagapan ang misfolded proteins by chaperone, ang gagawin usually ng katawan is it will do its best, okay? It will do its best para ma-degrade siya, okay? Para ma-degrade. So magkakaroon ng degradation, for instance, resulting to inactive peptide fragments, or okay na siya. Kaya lang, in certain situations, the body cannot cope up already with the triggers or problems. Kaya may mga pagkakataon na hindi sumasapat ang proteostasis, kaya nagkakaroon ng mga aggregates. And kapag nagkaroon ng aggregates, pag hindi ito na-manage, magkakaroon ng sakit. Kaya magkakaroon ng sakit. So para lang siyang homeostasis, dito ang tawag lang proteostasis. Everybody, can you follow? Okay, that's right. O kumusta ang reception sa Zoom? Mas maganda ba kaysa Google Meet? Alam na. So, eto na. Okay? Pinag-aralan ko kagabi eh. Meron palang wait lang ayan. Meron palang promo ang Zoom kapag ang account mo ay educational account. So, .edu.ph, parang ganyan. So, tinry ko, ayan, unlimited time tayo. Okay? So, alam na. Ito na lagi gagamitin. So, continue natin. Pwede na tayo mag-continue, guys. Okay, so in protein folding, we discussed that protein folding happens in milliseconds. So when we say milliseconds, sobrang bilis. Kaya we can expect that in a short period of time, there will be numerous proteins that will be produced. And we discussed also that the ribosomes are the ones that produce nascent polypeptides. However, such formation is not yet enough. Kasi pag sinabing nascent, may kwenta o wala pa sa katawan, wala pa siyang kwenta. Okay? Wala pa siyang pakinabang sa ating katawan. That's right. So increased concentration of proteins affect the kinetics of folding. So ibig sabihin, the many or the more proteins are being produced, the greater the rate of folding. Directly proportional siya. And please note that ang synthesis process held by ribosomes of nascent polypeptide would serve as determinant if ever ang protein ba ay magiging maganda or magiging beneficial sa atin, or ultimately magiging toxic or magkakaroon ng adverse effects. So kaya nakalagay dito, information needed to correct protein folding is contained in the primary structure. Ang point is, pag may mali sa primary sequencing ng polypeptide, susunod na dyan ang mga higher levels that could result ultimately to protein folding. Okay, protein folding. Now, ano ba yung pagkakatandaan nyo? Kung natatandaan kahapon anong pagkakatandaan nyo sa primary structure? Pag primary structure, ano ang characteristic bond na meron? Peptide bond lang, hindi ba? So, peptide bond. Ang peptide bond ba ay rotational or hindi? Hindi. Very good. Hindi siya rotational. Meron siyang partial double bond character. Dito ba natin ika-categorize ang amino acid sequence? Yes or no? Yung sequencing ng amino acids, yes. Okay, tandaan dito naka-categorize ang amino acid sequence. That's correct. So moving forward, we have different factors that could facilitate both in folding and refolding of proteins. So pag sinabing refolding, itatama kung ano ang maling folding to prevent diseases. So first, we have native conformation of a protein is said to be dynamical or thermodynamically favored. Pag sinabi nating thermodynamically favored, this tells us that the protein folding is said to be or is a process that happens in an orderly and also guided fashion. Again, this occurs in an orderly or guided fashion. Kaya nga nabanggit ko kanina, anong sabi ko sa proteostasis? Ano yung N na sinabi ko? None. Anong sinabi ko? Non-random. Okay, non-random siya. So it occurs in an orderly and also guided fashion. So that is synthesis, and then after synthesis, folding, and then after folding, degradation in case may problem. Next, folding is said to be modular. So pag sinabi naman nating modular, the events that happen, again, the events that happen is said to be step by step. Okay, step by step. So that is step by step. folding is modular. So in stage 1, there is synthesis nga, a synthesis, newly synthesized polypeptide folds, and then mapapataas ang level of structure niya, and then after that, dito na papasok ang proper regulation. Okay, proper regulation. In proper regulation, to maintain normality of the produced protein, it has to be realized that the exterior part of the protein being produced is where the hydrophobic ba or hydrophilic residues ang nakapwesto. Pag sa exterior, para normal yung protein, ano dapat ang nandito? Polar or non-polar na mga residues? Para masabi mong normal yung nagawang polypeptide or protein. Polar, very good. Pag sinabing polar, hydrophilic or phobic? Philic, very good. So hydrophilic siya. That is in the exterior, And then in the interior naman, again, in the interior, ano dapat ang nasa loob ng produced or folded polypeptide or protein? That is non-polar. That is hydrophobic. Okay, hydrophobic dapat siya. I repeat, exterior, polar or hydrophilic, and then interior, non-polar and phobic. That's why you have to familiarize yourselves once more on the amino acids na may polar and non-polar side chains na tinakal natin ng lesson 1. Until such time that native conformation will then be achieved. Ito ba yung ultimate goal na folding? Yes. Kasi para ma-render nga ang protein na functional. Okay, na functional. Can we proceed on this slide, guys? Okay, so thank you. Moving on, let's have auxiliary proteins that assist folding. Remember, folding process has to be regulated because not all the time the product or the protein that will eventually be produced would be good. Kaya kailangan may nag-a-assist. Majority of the assistants are the chaperones as we discussed earlier. However, there are miscellaneous depending on their target protein such as protein disulfide isomerases and proline cis trans isomerase. So first, we have chaperones. So discuss natin what are chaperones. Di ba ginagamit din natin ang term na chaperones pag may kasama ka? O parang ganon ang chaperone na protein. Ang ibig sabihin ng chaperones, assistant sila ng mga proteins habang finofold. Kaya chaperones ang tawag. These are specialized group of proteins that are required for proper folding, for proper folding of many proteins or many species of proteins. So these are also known as polypeptide chain binding proteins. Okay, so they are so-called as polypeptide chain binding proteins, and they can also act as catalysts. So when we say catalysts, they can actually hasten, the chemical reactions involved in protein folding. Anong ibig sabihin ng they can hasten? Pinapabilis o pinapabagal nila? Pag-hasten? Pinapabilis, that's good. So they can hasten, pinabibilis nila ang rate of chemical reaction. So that's the role of chaperones. Now, ang chaperones ay martir. Bakit siya martir? Kasi ang chaperones, sumasama lang siya sa journey pero hindi sa final destination. Anong ibig sabihin yan? Pag kailangan siya, nakasunod siya. O sasamahan kita protein, kailangan tama ang folding mo. Pero pag na-achieve na ng protein ang correct native conformation, aalis na siya. Kasi feeling ng chaperones, hindi na siya kailangan. Okay? Kaya nakalagay dyan, do not form part of the final structure ng protein. So walang hugot doon para lang magets nyo. Okay? So it only assists during the journey, during the journey, yes. Pero pag narating ang final destination, feeling niya, hindi na siya kailangan. O yung mga chaperones dyan, mag-isip-isip na kayo ha. Okay, next. After natin sa info na do not form the part of the final structure, they also suppress non-productive interactions by binding transiently. Kaya nga ang term natin ay during the process or journey lang kasi transient lang ang binding. And they were first identified as heat shock proteins. By the way, nasusundan pa ba ako? Okay. So, they were first identified as heat shock proteins. Why? Based on experiment before, tumataas ang concentration ng chaperones, tumataas ang concentration ng chaperones kapag ang temperature ay elevated sa cell, during elevated temperature. Again, ang heat shock proteins or chaperones are said to be abundant in cells that are being stressed by high temperature environment. Kaya heat shock. Question, ang heat ba ay isang trigger for protein destruction? Yes. Okay? Kaya hanggat may iwasan ang destruction ng protein, chaperones are acting. Kaya tumataas sila at high temperature environment. That's why they were first identified as heat shock proteins. And then, heat shock protein expression is elevated. Na-discuss ko na yan. Elevated when cells are grown in higher than normal temperature. And they act through ATP-dependent mechanism. So, na-discuss natin yung figure na to kanina. You can see here, apparently, that there are chaperones that act in almost every step of the way. Because ang ultimate goal ng chaperone, ulitin natin, the bottom line is to prevent aggregation or misfolding of proteins. So meron tayong iba't ibang klase ng chaperones. Again, meron tayong iba't ibang klase ng chaperones. First stop, we have the heat shock protein 70. Ang heat shock protein 70 is often found in the cytoplasm, in the endoplasmic reticulum, in the chloroplast, and also in the mitochondria. So this type of HSP or chaperone binds to regions of unfolded polypeptides such that the hydrophobic residues will be placed interior. Will be placed interior. Ito ba ang magandang outcome kapag hydrophobic ay nasa loob? Magandang outcome ba siya pag ang hydrophobic ay nasa loob ng protein? Yes. So yan ang function ng HSP70. Usually ang inaatake nila yung mga polypeptides na maraming hydrophobic residues. Kasi pag maraming hydrophobic, mataas ang chance na sumulpot sa labas. Masama yun. Kailangan itago as much as possible. So it blocks folding of proteins that should be remain unfolded and facilitates quaternary assembly of oligomeric proteins. Another type, we have chaperonins. Chaperonins are so-called as cage-like. These are so-called as cage-like chaperones. I repeat, chaperonins are so-called as cage-like chaperones. That's why, as you can see on the picture, they look like cages. Okay? They look like cages. So magkaiba ang specific term sa chaperonin depending kung prokaryote ang cell or eukaryote. Kapag prokaryote, it's GROW-EL. Pag naman eukaryote, it's HSP60. Okay? HSP60. So just take note the description of these types of chaperonin. So I repeat, cage-like chaperones, we have two types. For prokaryotes, we have GROW-EL. GROW-EL talaga yung pangalan niya. And then for eukaryotes, we have HSP60, okay? Heat shock protein 60. So take it all together, that's the end of the first auxiliary protein. Ang first auxiliary protein natin ay chaperones that assist in folding. Now, meron ba tayong nabanggit na mga miscellaneous kanina? Again, meron ba tayong nabanggit na mga miscellaneous kanina? Yes or no? Check your notes. May miscellaneous ba tayong auxiliary proteins? Di ba majority ay chaperones. So meron tayo mga miscellaneous. Ano yung mga miscellaneous? Meron ba? Choppy ba ako? Or di lang talaga kayo nagre-reply? Or nawala ba ako? Hello? Nawala siguro ako. Meron. O ba't di kayo nasagot? Ba't di kayo nasagot, ha? Pag kasi hindi nasagot, feeling ko, nag-iisip. Oo nga naman. Huwag mo kaming istorbohin, sir, nag-iisip kami. Kung paano kami mananatiling gising sa oras na to. Nag-iisip pa kami. Balikan na lang natin yung slide, ha? Ito yung auxiliary proteins. Naalala nyo to kanina. Di ba tatlo? Majority ang chaperones. Ito yung miscellaneous. Okay, ito yung miscellaneous. Okay na? Naalala na? Okay. So, punta na tayo sa miscellaneous. Second, we have protein disulfide isomerases. So protein disulfide isomerases stabilizes, again, it stabilizes the tertiary and quaternary structures, and usually it eliminates folding of intermediate with, so it eliminates folding of intermediates with inappropriate, nandito na pala, with inappropriate disulfide crosslinks. So with that being said, again, with that being said, we can realize that nakafocus ang protein disulfide isomerase sa mga residues na may disulfide linkages. Ano ba yung may mga disulfide linkages? Yung cysteine residues. Ay yung mga polypeptides na may cysteine residues. Because it is the one responsible for disulfide bond formation. Okay? Next, we also have proline cis-trans isomerases. So these enzymes are also known as cyclophilins. Again, these are also known as cyclophilins. And these are usually found, or they actually target the polypeptide na may X-pro peptide bond. Again, tinatarget nila ang may X and pro peptide bond. Anong meaning ng X-pro? Pro is proline. Okay, so this refers to the amino acid proline. And then X, ang ibig sabihin ng X is any amino acid. Okay, any amino acid. So anong point niyan? Again, what's the point of this X-pro peptide bond? In short, ang tinatarget ng proline cis-trans isomerase ay yung mga polypeptide na may peptide bond na katabi ng proline. Lahat ng katabi ng proline. Okay? Kahit anong amino acid pa yan, basta katabi ng proline. Doon nag-a-act ang proline cis-trans isomerase. So this enzyme catalyzes isomerization of the bond from being trans to cis. Okay? From being trans to cis. Kaya ang term natin ay cis-trans isomerase. Okay? Next. So overall, we are done discussing auxiliary proteins. So going back for the auxiliary proteins that assist in folding, we have three. The major ones are, ano yung majority? Anong tawag sa kanila? Chaperones, right? And then for the assistant, or let's say miscellaneous, we have disulfide and also cis-trans. Okay, so take note of that. Kapag pag proline cis-trans isomerase, X-pro. Kapag disulfide, that is cysteine residues. So yun yung palatandaan natin. And then for the last, a last factor affecting protein folding, this is a dynamic process. So when we say dynamic process, multiple processes occur so as to maintain proper folding of proteins. That's why we discussed that chaperones do their best to be able to rescue the unfolded proteins, or potentially yung mga magmi-misfold. They are always to the rescue. Okay, yun yung function ng chaperones. And then for glutathione naman, accordingly, this is the one that reduces inappropriate disulfide bonds that could be seen in the polypeptide, particularly those that are placed exterior. Okay, those that are placed exterior. So these are the factors. Okay, these are the factors that could affect protein folding process. Can you follow? What about the others? How are you? Are you still awake? I cannot see you. Don't have video eh. Pero kita ko kayo ngayon. I mean, kita ko yung video grid. Kaya lang, wala kayong mga mukha. So, hindi ko alam kung kumusta na kayo. Ha? Pwede na tayo mag-continue? Okay. Nice. Remember, so far we were able to discuss proteostasis, particularly the process of protein folding. So kung meron tayong term na protein folding na diniscuss natin ngayon lang, ang susunod na slide ay may kinalaman naman sa unfolding. So pag sinabi nating unfolding, again, pag sinabi naman nating unfolding, ito ay kabaliktaran. That is, yung protein na functional ay nagiging non-functional. Okay? So tandaan niya na. So kung kanina diniscuss natin ang pagiging pag-convert ng protein para maging functional. Ngayon, yung susunod na slide na i-discuss ko, may kinalaman naman to sa kabaliktarang pangyayari, wherein ang functional na protein gagawin nating non-functional. Nagets yung key concept? Nagets yung sense ng next part ng discussion? Okay, so that's why ang next topic ay may kinalaman sa term called protein denaturation. In protein denaturation, we tend to disrupt the structure of the protein such that the biological action of the protein will be removed. Mawawala siya, will be omitted. Kaya sinabi ko na may kinalaman ito sa pag-convert ng isang functional protein to a non-functional. So therefore, let me ask you, with this being said, during protein denaturation, pinapataas ba natin ang level ng protein structure o pinapababa natin ang level ng protein structure? Taas o baba? Very good. Pinapababa natin ang level ng protein structure. So kung dati kapag proteostasis from primary, secondary hanggang quaternary, ito, from being quaternary, pabababain natin to tertiary to secondary until such time na magiging primary siya na ang tendency, wala nang function sa katawan. Ganon ang protein denaturation. Nagets ba ang principle ng denaturation? Okay, so in protein denaturation, again, we tend to reduce folding of protein such that the quaternary structure, let's say, ay napupunta into its primary level, rendering the protein non-functional. However, again, tandaan, once primary structure has been reached, this is now the lowest level. Ito na lang ang pwedeng pinakamababang kahinatnan pag ang protein ay na-denature. What do I mean by that? Again, anong ibig sabihin natin sa lowest level? Pag sinabi nating lowest level, pag na-denature ang mataas na klase ng protein, ang pinakapangit, kumbaga, na structure na lang niya is sequence of amino acid residues. So sequence na lang polypeptide. But take note, we cannot anymore separate all the residues through denaturation. Okay? So hindi kaya ng denaturation ang separation ng mga residues. Instead, ang magbe-break ng peptide bond ay hydrolysis. Okay? So ang magbe-break ng peptide bond ay hydrolysis. So I repeat, hydrolysis is the process of breaking peptide bonds. Siya lang ang makakapaghiwalay sa mga residues. Denaturation is limited hanggang sa sequence lang. Did you get my point? Okay, very good. That's why you can see here na yung isang protein sa figure sa baba, yung isang protein, complex protein na nag-curl na somehow, so may proper folding na, during denaturation, nawala yung proper folding niya. So as simple as that. Okay? So as simple as that. Next. So ito applicable sa lab. Ang susunod na question is, makinig, Sir, ano mga possible factors na pwedeng magpa-denature sa protein? Of course, usually external factor yon. Halimbawa, folded na yung protein, functional na yung protein, may external stimulus. May adverse stimulus from the outside kung saan na-expose ang protein. Doon siya madedenature. Ang pinaka-common, exposure to temperature. For example, pag sobrang taas ang temperature o kaya changes in pH. Tapos na kayo sa lab sa ganitong mga topics sa lab? Or not yet? Tapos na. Pero mas nakilala nyo ba kung anong nangyayari sa denaturation? Mas nag-experiment kayo. Pero mas naintindihan nyo yung principle niya, nagiging non-functional yung protein. Okay? Nawawalan siya ng kwenta. Parang ganon. So, ano yung mga possible factors, possible triggers para masira ang protein from its being functional? So, meron tayong iba't ibang causes. Check the next slide. May table, di ba? Yung sagot sa blank na to, yung nasa table sa next slide. So just take note the agents that can cause protein denaturation such as heat, UV radiation, acids and bases, urea, and so on. So it's your responsibility to read this table and also observe the effect. So halimbawa, magbigay ako representative ha. Halimbawa, heat. Ang heat, sinisira niya ang hydrogen bonds. Question, ang hydrogen bond ba ay importanteng force of attraction para mag-fold? Para mag-fold o mag-bend yung mga sequence? Yes or no? Ulitin natin for the benefit of everyone. Ang heat daw, sinisira niya ang hydrogen bond. Importante ba ang hydrogen bond para mag-fold ang protein o ang peptide? Yes. Di ba ang secondary structure na discuss natin may hydrogen bond? So pinapababa talaga natin. Okay? So very good. Now, ang question dito is, balik tayo, ang question dito is, ang protein denaturation ba ay reversible or irreversible? Take note, most of the time, it's irreversible. Okay? Irreversible. So, irreversible meaning, once the protein has been denatured, yun na siya. Okay? Babay na siya. Unless, meron tayong special process or special treatment na gagawin through renaturation, wherein maibabalik natin siya into native conformation, but it's quite difficult. Okay? Pero generally, you can classify protein denaturation as irreversible. Okay? Do you understand so far? Okay. And then, guys, pakibasa silently yung effect na nakalagay dyan. Yung effect ba, naintindihan na ba yung ibig sabihin nito based on my discussion? Based on my discussion, unfolding and disorganization not accompanied by hydrolysis of peptide bond. Okay? So, ang hydrolysis lang ang makakasira ng peptide bonds. So, next. Ito na yon, nasabi ko, commonly, irreversible siya. Kaya nga nakalagay sa reversible rare cases. Okay? Yun lang ang tandaan nyo sa part na yan. Overall, naintindihan ang protein denaturation. How about the others? Are you well? Okay pa kayo? Ayan. Recap tayo. Sa second part on lesson 2, we were able to discuss protein folding process and protein denaturation. Now, to trigger your minds sa next discussion natin sa lesson 2, hindi ba ang proteostasis ay mahalaga para ang protein na magagawa hindi magkukos ng disease? Pero syempre, may mga pagkakataon that the body cannot anymore regulate proper folding. That's why diseases happen. In connection to that, let us tackle the different pathologic consequences of protein conformation perturbation or meaning misfolding of proteins. So meron tayong iba't ibang klase ng sakit that can be attributed by misfolding of proteins. First stop, we have the prion diseases. Actually, pag sinabi nating prion, meron siyang ibig sabihin. Ang ibig sabihin natin ng prion ay proteinaceous. Again, this means proteinaceous infections only. Again, when we say prion, it means proteinaceous infections only. Kaya prions. Okay? Proteinaceous, prion. Okay? Nagets niyo ba't prion ang tawag? Para siyang abbreviation. So regarding prion diseases, these are transmissible spongiform encephalopathies wherein the primary target is the brain. Brain. Kaya nga encephalopathy ang tawag. So this is actually fatal. Now, may mga variations tayo sa tawag sa mga prion diseases depending on the organism being affected. Okay? Depending on the organism being affected. So halimbawa, kasali sa prion disease ang Creutzfeldt-Jakob disease, which affects the humans, scabies affecting the sheep. Okay? And then we have mad cow disease in cattle. Ang Alzheimer's disease ay separate form siya pero prion din siya. Okay? Pero prion disease din siya. Theoretically or hypothetically, let's say. Now, regarding Creutzfeldt scabies, nasusundan pa ba ako? Nasusundan? Creutzfeldt scabies and BSE or bovine spongiform, same manifestation but it only differs which organism is affected. So halimbawa, ikaw, sinabihan ka, Nako, si Pedro may scabies. Wag kang papayag kasi hindi ka tupa, tao ka, okay? Tao ka. So parang gano'n. Hindi ka pwedeng sabi, "Ay, may mad cow siya." Hindi ka baka, okay? Hindi ka baka. Dapat ang tawag sa condition mo, dapat ang tawag sa condition mo ay social dapat. Creutzfeldt-Jacob, okay? Creutzfeldt-Jacob ang tawag diyan. Naintindihan? Naintindihan? Okay. So regarding prion disease, ano ang reason bakit may prion disease? So may kinalaman ito, ang bottom line lang nito, may kinalaman sa misfolded proteins. Ngayon, anong klaseng misfolded protein ang involved? Again, anong klaseng misfolded proteins ang involved? Para magets ito, i-divide natin yung discussion into two. We have the first and the second. In the left, again, in the left, the first one is the normal. Inside our body, again, inside our body, we have the normal protein called PRPC. Again, we have the normal protein present called the PRPC. So when we say PRPC, it means, again, when we say PRPC, it means cellular prion related protein. Again, pag sinabi natin PRPC, this is cellular prion-related protein. Wag kayong magpapadala sa pangalan kahit may prion na term, normal protein siya sa katawan. Pero related kasi siya, okay? So, prion-related protein. Let's talk about the structure of the prion-related protein. It says on the slide, the normal protein, PRPC, has a secondary structure containing alpha helices. Okay, alpha helices. And then easily soluble siya, ay easily soluble, and can actually be degraded or hydrolyzed by proteases. Okay, by proteases. So yan ang characteristics ng PRPC. Now, if proteostasis is not well maintained, chances are this normal PRPC could become PRPSC. Okay, PRPSC. Ang PRPSC, ito naman yung abnormal, okay, abnormal prion-related protein. Abnormal siya, kaya nakalagay abnormal because it is the one that can cause encephalopathy. As we check the structure, makinig, as we check the structure, instead of having alpha helices, ang meron beta sheets. Okay, ang meron ay beta sheets. Magkaiba ba yung alpha at beta conformation sa secondary structure? Yes. Okay? Kaya yung structure ang magde-determine kung siya ba ay good or bad. Now, unlike the normal protein, this is insoluble, yung PRPSC, insoluble. So pag insoluble, kapag indigestible siya by certain solvents, anong ine-expect nyo? Sino ang nasa exterior? Sino yung nasa exterior? Is it the polar or the hydrophobic? Kapag hirap tunawin, usually anong maiisip natin? Ano yung nasa exterior? Hydrophobic or hydrophilic? Hydrophobic. Question, pag ang nasa exterior ay hydrophobic, is it good or bad? Bad. Okay, kaya problematic siya. Are you learning so far? Okay, so that is for PRPSC. O yun lang tandaan nyo yung pinagkaiba nila. Okay? So yan yung prion diseases. So next, since nabanggit natin na ang Alzheimer's ay isang halimbawa, hypothetically, ng isang prion disease, ipaliwanag natin anong problem sa Alzheimer's. Familiar kayo sa Alzheimer's, right? Ang Alzheimer's ay isang neurodegenerative disorder caused also by misfolding of proteins. At ang tawag natin, everybody, sa misfolded protein ay beta amyloid. Okay, beta amyloid. Now, ano ba yung beta amyloid? Ang beta amyloid ay isang misfolded protein that accumulates, tumataas siya in people with Alzheimer's disease. Okay? In people with Alzheimer's disease. And also it was found out that in Alzheimer's disease, there is also low level of the neurotransmitter acetylcholine. Again, the problems in Alzheimer's based on studies, mataas ang misfolded beta amyloid at mababa ang acetylcholine. Nakikinig pa ba? Okay. So, with these two, so far, may mga gamot tayo available in the market that that increase acetylcholine levels. What I'm trying to say is may gamot pang Alzheimer's by targeting acetylcholine. Okay? By increasing acetylcholine. So check tayo dito kasi may drugs available. Pero, makinig, pero ang problema is, again, sa problemang beta amyloid na mataas, wala pa tayong gamot available in the market. Okay? Wala pa tayong available in the market. So in connection to that, i-discuss natin ang simple step kung paano ba nabubuo ang beta amyloid. So ang basic step huwag nyo nang pansinin to ha, huwag nyo nang pansinin. Doon tayo mag-focus sa beta amyloid, okay? So doon tayo mag-focus sa beta amyloid. Ang beta amyloid ay nanggagaling sa tinatawag na amyloid precursor protein. Usually, ang symbol ng protein, para lang aware kayo, Chon. Kasi ang mga proteins, may carbon, may hydrogen, may oxygen, may nitrogen. Okay? So, amyloid precursor protein. Now, ang amyloid precursor protein, hindi ko sure, baka di na ako nasusundan na, ulitin natin. Amyloid precursor protein ay nagiging beta amyloid through an enzyme. Okay? So, may enzyme na kailangan. Ang tawag sa enzyme na yon ay beta secretase. So, ang tawag sa enzyme na yon, hindi masyado malinaw, pero ito yung spelling, beta secretase. Yan, beta secretase. So, this enzyme is responsible for the conversion of amyloid precursor protein into beta amyloid. So, if that is the case, please listen. If that is the case, please answer my question. If you are a scientist, and you want to discover a drug that would lower beta amyloid, do you think blocking the enzyme beta secretase is a good goal or is an ideal goal? Yes or no? Yes, okay, very good. In Tagalog, kung ikaw ay magdi-discover ng gamot pang Alzheimer's na magpapababa ng misfolded beta amyloid, pwede kang mag-discover na nagbablock ng beta secretase. Kaya marami tayong thesis or thesis, marami tayong mga researches na kinoconduct na ang goal ay targetin ang beta secretase, pwedeng gamitin sa Alzheimer's. Kaya pag nagbasa kayo ng mga studies at nakita nyo na beta secretase inhibition, yung action ng compound, pwede mong isipin na ang gustong iparating ng study pang anong sakit? Alzheimer's. Kasi wala pang nagta-target ng beta amyloid na established worldwide. Okay? Gets ba? Gets? Okay, very good. Next example, hindi na to prion disease ha, hindi na to prion, pero epekto ng misfolded protein. Okay, epekto ng misfolded protein. Next, we have beta thalassemia. Beta thalassemia is a collective term pertaining to genetic defects that impair the production of one subunit of hemoglobin. Sa susunod na lesson, which will be discussed also today, ang hemoglobin ay nagke-carry o nagta-transport ng oxygen. So mamaya maiintindihan. Di ba no elementary tayo at high school, ang alam lang natin, hemoglobin transports oxygen. Pero sa biochem nyo maiintindihan paano yun nangyayari. Okay? So explain natin yan mamaya. And ang problem sa beta thalassemia, since defective ang hemoglobin, may problems sa oxygen transport. So that is because of the presence of ASHP, causing cytotoxicity of developing red blood cells. So that is beta thalassemia. We also have sickle cell anemia. Again, we also have sickle cell anemia. Ganito kasimple ang sickle cell anemia. Ang sickle cell anemia, sa polypeptide sequence, yung glutamic acid na residue, napalitan ng valine. Okay, napalitan ng valine. So I repeat, ang problem sa sickle cell anemia, yung protein na sequence, yung polypeptide sequence sa hemoglobin, instead of glutamic acid, ang pumalit valine. O di ba, mapapansin nyo, by the way, nakakasunod pa ba? Nakakasunod pa? Ha? Okay pa kayo dyan? Okay. So, kung mapapansin nyo, yung residue, isang pirasong residue lang ang nagbago. Yung glutamic acid naging valine. Primary level lang yon. Nagkamali ng isa, domino effect na siya hanggang highest level. So kung mapapansin sa picture, ang normal red blood cell na dapat glutamic acid ang isang residue na ganito ang itsura, biconcave dapat, naging crescent shape siya dahil lang sa pagpalit ng valine. O gano'n na nangyari. Now, anong implikasyon pag naging crescent-like shaped yung itsura ng red blood cell? Hindi na siya makakapag-carry ng oxygen. Pag hindi siya nakapag-carry ng oxygen, magiging anemic yung tao. Kaya sickle cell anemia ang tawag. Okay? Gets ba? Okay. Ano dapat ang normal na hemoglobin? Anong sequence? Glutamic or valine? Ano ang glutamic acid or valine? Glutamic acid, good. Tawag dito, ano yung napalit? Valine. Okay, so ito yon. Yeah, okay, ito yon. So next, I think this is the last slide for this lesson. So other important proteins, blood sugar level in the body is being regulated by a protein, and that is a hormone called insulin. Okay, so that is insulin. So take note of the effects of insulin. Don't worry, may specific module tayo na i-discuss natin ang insulin sa metabolism. Doon natin siya i-discuss. So sa ngayon, tandaan nyo, ang insulin can cause cellular glucose uptake resulting to hypoglycemia. Okay, resulting to hypoglycemia.